Okay, let's unpack this.
Right.
Welcome to our deep dive. We're actually drawing directly from the transcript and the class notes from FIT 678.
Yep, that's the statistical genetics course taught by Professor Guilherme da Silva Pereira.
Exactly, over at the Department of Agronomy at the Federal University of Visotsa.
Mm-hmm.
And today we are wading into, well, it sounds complex, multi-trait, multi-environment QTL mapping,
Mm-hmm.
or MTME.
It is a mouthful, and yeah, it can get complex, but it's incredibly important stuff we're talking about.
So the mission really is to understand why. Why is looking at multiple traits, maybe across different environments, why is doing that together so crucial?
Right. Why is that joint analysis the key, to unlocking the genetics behind complex characteristics?
Because I guess most real research isn't just one trait in one place, is it?
Almost never. I mean, think about plant breeding. You care about yield, sure, but also disease resistance, maybe quality traits.
Or human health, you might measure body fat,
Yes.
blood pressure, cholesterol, maybe response to a drug.
Exactly. And you often measure these things in different conditions, different locations, different years, different populations.
And these things aren't isolated. Traits are often correlated, right? Or
Yeah,
they behave differently depending on the conditions.
precisely. And the challenge isn't just seeing that correlation, it's understanding the genetic reason behind it.
So if you've got all this data, multiple traits, multiple environments, what's the first thing someone might try? The simple way.
Well, the course notes call it the naive approach. Basically, you just run a separate analysis for every single trait in every single environment.
Okay, so like dozens of separate QTL mapping runs.
Potentially, yeah.
Yeah.
Then you just sort of, well, look at all the results side by side. See if any QTLs happen to pop up in roughly the same genetic region.
Okay, I can see doing that. But what's the big drawback?
Mhm.
If you see a peak for yield and one for resistance near each other, doesn't that tell you
It
something?
tells you something is going on there, yeah. But it doesn't tell you why they're co-locating. You can't statistically distinguish the cause. Meaning, is it one gene with effects on both traits? That's what we call pleiotropy. Or is it actually two distinct genes, one for yield, one for resistance, that just happen to be very close together on the chromosome and are usually inherited together? That's linkage.
Ah, and the separate analyses can't tell those two scenarios apart formally.
Nope. You lose that crucial detail about the underlying genetic architecture. You're just guessing based on proximity.
Okay, so that's where MTME comes in, the joint analysis.
Exactly. Models like the MTMI multi-trait multiple interval mapping mentioned in the notes, they look at everything simultaneously in a multivariate framework.
And the concept that lets you interpret these models is genetic correlation.
That's the key. For multiple traits measured together, genetic correlation quantifies how much of the genetic influence on one trait is shared with another.
High correlation suggests pleiotropy or linkage.
It could be either, but the joint model gives you tools to test which is more likely.
And what about genetic correlation for the same trait but in different environments?
Then it measures genotype by environment interaction, or GXE. If the genetic correlation is high, say close to 1, between yield in environment A and B, the genes are acting similarly.
But if it's low or even negative,
That means there's significant GXE. The genetic control of the trait is different in those two environments. Some genes might be important in A, but not B, or vice versa, or even have opposite effects.
right? So what are the concrete advantages of doing this joint MTME analysis?
Well, first off, power, statistical power. By pooling information across traits and environments, you have a better chance of detecting QTLs, especially ones with smaller effects that might be missed otherwise.
Makes sense, combining evidence. What else?
Better resolution, potentially.
Yeah.
You might be able to narrow down the location of the QTL more precisely on the chromosome.
Okay, power resolution.
And I think this is the big one, formal hypothesis testing. You can use statistical tests built into the model to explicitly ask those questions we couldn't answer before.
Like, is this pattern best explained by one pleiotropic QTL or two linked QTLs?
Exactly. Or does the effect of this specific QTL significantly change across these environments? Is the GXE statistically significant for this locus?
The source material had some real-world examples, didn't it? Like fruit flies.
Yeah, studies on morphometric traits in Drosophila, and also maize, looking at drought tolerance and yield under different water conditions.
And those examples actually showed the joint analysis finding QTLs that the separate ones missed.
They did. You could see clear cases where the increased power made a difference. And in the maize example, you could visualize the GXE plots showing how the estimated effect of a single QTL changed dramatically depending on the environment.
This all sounds incredibly powerful, but I'm guessing analyzing all that data together,
Yeah.
it's not easy.
No, it can get computationally very intensive. Think about all the parameters you need to estimate in these joint models. Genetic variances, covariances, QTL effects for every trait in every environment.
So it takes a lot of computing power and time.
Definitely. It can be slow, especially with many traits or many environments. Sometimes you might need to make compromises, like grouping similar traits or environments to make the analysis feasible.
So practical hurdles exist, but the payoff is.
The payoff is a much, much deeper, more accurate understanding of the genetic architecture of these complex traits.
And knowing that architecture matters, like your example of yield and disease resistance.
Absolutely. Knowing why they're correlated, true pleiotropy, where one gene affects both, maybe in opposing ways, or just tight linkage, that has huge implications.
How so?
Well, if it's pleiotropy with an unfavorable correlation, breeding for both high yield and high resistance might be fundamentally difficult, biologically limited.
Okay.
But if it's linkage, even tight linkage, well, linkages can be broken by recombination eventually. It might be hard, you might need large populations, but it suggests there's a possibility of finding that rare combination of high yield and high resistance alleles together.
So understanding that precise genetic relationship directly informs the strategy you'd use.
It absolutely should, which, raises a broader question maybe for people to think about.
Yeah.
How much does really nailing down these genetic relationships using approaches like MTME actually change the game? How fundamentally does it alter the strategies we use in plant breeding or animal breeding, or even understanding complex human diseases to actually make progress on these multifaceted problems?